Okay, so let's learn something. Um, so who is so done with the semester? I am too. How are we gonna get through this next week and a half or so? Um, the good news is we are so far ahead of where I was this time last year that I'll actually get to teach you guys about apoptosis in its entirety, which is actually very exciting because last year my daughter got sick and ended up in the hospital. I had to cancel class at the end of the semester, and it was a total bummer. Well, it's a bummer she was sick, but she was fine. But it was a bummer because I taught you guys all about cell cycle and cell growth, and then didn't teach you anything about apoptosis. Not you, the former you that sat here. Um, so this year, no one gets sick, and you'll learn about apoptosis. Okay, so what did we do the other day? So we've been learning about the cell cycle, and we started just with the basics and the key players and what's going on. Right, there are different phases of the cell cycle. There are different checkpoints. Checkpoints are there to make sure that you don't proceed if things aren't right. Right, if your DNA is damaged, if your chromosomes aren't attached to your spindle, mitotic spindle, those kind of things, that you stop and you do not proceed with the cell cycle. Otherwise, you get bad things. Um, and then we learned about the key players, cyclins, CDKs, and how they're regulated, right, with phosphorylation and CKIs and degradation of cyclins and other proteins. We learned also about these ubiquitin ligases that degrade things. We learned about S phase and how SCDKs, remember wherever we're learning about CDKs, you have to think it's a kinase that's phosphorylating something. What is it phosphorylating? How is it affecting that thing's activity? So in S phase, it's all about regulating, well, not all about, but we learned about regulating pre-RC formation and disassembly and how that's involved in initiation of, I feel like really no one's here today. People are giving up. Okay. How it's involved in regulation of DNA replication initiation. And you want to think about CDKs and APC when you're studying this. What is the SCDK doing? What is APC doing? How does this affect initiation of DNA replication? Then we started talking about regulation of M phase and MCDK cyclin activation, distracting, and targets. Right, so what are some targets, right? What are some targets of MCDKs? Do you guys remember this? Just think about it. MCDKs are kinases. They must be phosphorylating something. This is somehow involved in activation of mitosis. What might an MCDK be phosphorylating? Make something up. What's important for mitosis? Mitotic spindle, yay! What else? What works with mitotic spindles to separate chromosomes? Does that work with mitotic? See, you were just like you had it in there. That's an important one, but not with the mitotic spindle. What works with mitotic spindle in order to separate chromosomes? When we learned about the cytoskeleton, what works with? Huh? Microtubules, mitotic spindle. Okay. What walks along? Motor proteins. Remember, motor proteins are. You can have a mitotic spindle, but if you don't have motor proteins, are you going to have chromosome segregation? No. Right? You need motor proteins to bind. There are motor proteins that bind to the cell, like the cell cortex, and help to position the spindle. There are motor proteins that bind to chromosomes and help to line them up in metaphase, right? So one of the targets of MCDKs are motor proteins, remember? Regulation of kinesins by phosphorylation. Put it all together. Remember that figure? The thing was folded. There's something folded. No, that was myosin. There was another figure. Okay. Anyways, so that's where we stopped. We were talking about the role of MCDKs in mitotic spindle assembly and function. Okay, so this is where we'll pick up. So of course, you need to assemble your mitotic spindle, and you need to line up your chromosomes in metaphase. But a really important thing that needs to happen is you need to attach your mitotic spindle to your chromosomes, right? And we talked about this, how the microtubule is attached to the kinetochore, and there's this ring structure, and then there's catastrophe, and that shrinks the microtubules, and they separate the chromosomes, and that's what happens. So you can imagine that if you have incorrect attachment of your microtubules to your chromosomes, it would be bad, right? What do I mean by incorrect attachment? What does that mean to you guys? Yes?
Like if, like the picture says, like if they're not attached, like both are attached at the same point of the kinetochore, both of them are attached to one point, then it's not gonna like pull like correctly. It's gonna break the DNA.
Right. So if both mitotic spindles are attached to the same point or the wrong points, or just incorrectly. So if you look at this figure, you've got here just one pole of the spindle, right? One side is attached, the other side's not attached at all. Here you have two sides attached to one side, and this is also not correct. Here you have both poles, right, attached to one side. And then here you have the correct attachment. And so you don't want any of these things to happen because, like Ashley said, you could end up breaking your chromosomes. And what else could you end up doing? What if you segregate with this one? What's going to happen? What's going to be your resulting cell? Like, what's it going to look like? How many chromosomes will it have? Too many, too little, something like that, depending on which cell you're looking at, right? So it's really important that those sister chromatids, one goes in each direction, and that's why it's so important to attach your mitotic spindle correctly. Okay, so that's this checkpoint. That's one of these checkpoints that we've talked about that prevents metaphase to anaphase transition. It's very much involved with this being correct. So there's a kinase called Aurora B, and it senses whether or not the tension is correct. So there's going to be tension if you've got mitotic spindle microtubules attached to kinetochores, right? And so you can imagine that the tension here is going to be different from the tension here, right? Because one's not attached. And here they're attached equally on both sides. It's not completely, maybe it is, but not known by me exactly how that tension is sensed, but it does sense tension in some way. And it will phosphorylate the microtubule attachment site if the tension is not correct. And that will decrease the affinity of the microtubule for the kinetochore. So it basically causes it to unattach, which is good, right? If it's not attached correctly, you want it to detach. And that way it can try again. And so that's what happens. Any questions about that concept? Why it's important? Why we care? Okay. Okay, so you go through all this stuff really to get to this point, right? You line your chromosomes up at metaphase, and then anaphase is where you get separation, and this is triggered by APC. So APC is activated by MCDK. Someone emailed me and was like, where did you find that? Why is that the answer? It's in there. It's in there. There's actually phosphorylation that allows CDC20 to interact with APC, and that's what activates phosphorylation by MCDK. That's what activates APC in mitosis. And APC is going to degrade a whole bunch of things, one of which is securin. That's securin. So there's this enzyme complex called securin and separase. And separase is the actual enzyme, and it's kept inactive by being bound to securin. And once securin is degraded by APC, it allows separase to be active and go on and cleave cohesin, which is this protein complex holding together your sister chromatids. And that allows separation. So without that, you would never have proper segregation of chromosomes. So you know now that APC degrades securin. You know that it degrades MCDK, degrades other things. So how does activation of APC by MCDK create a negative feedback loop, right? So MCDK activates APC, and then also APC degrades other things. What does APC degrade that causes MCDK to create a negative feedback loop? Negative feedback means it turns itself off. So what does APC degrade that I just kind of told you? Other than securin. Right, who said that? Yeah, M-cyclin, right? So M-cyclin phosphorylates, activates APC, and then degrades itself. And so that's an important concept to remember, because towards the end of mitosis, you do need M-cyclin degradation. You cannot exit mitosis without degradation of M-cyclin and turning off of MCDK.
You said APC is also degrading M-cyclin?
Mm-hmm. Yep, APC degrades M-cyclin. So M-cyclin CDK complex phosphorylates and activates APC. That allows separase to become active and to degrade and to cleave cohesin, but also it degrades M-cyclin. And so M-cyclin and MCDK turns itself off by targeting its cyclin for degradation. And that's required for exit from mitosis. Okay, any questions about that? So remember, it's all about this up and down of these cyclins. So every time you're exiting a phase of the cell cycle, you need to think to yourself, are the appropriate things being degraded and are the appropriate things being turned off? Okay, so let's go back to spindle assembly checkpoint, right? So this is the attachment of the mitotic spindle to the chromosomes. If it's not correct, then this checkpoint is activated. The purpose of activation of a checkpoint is to stop the cell cycle, okay? So you need to think to yourself, well, how does this work, right? Oh, well, Dr. Ehrlich will tell you. Basically, MAD2 is a protein that's going to bind to unattached kinetochores. So in this picture, you can see your microtubules, which in this case is your mitotic spindle, right, are green. Your DNA is blue, and MAD2 is red. And so you can see your spindles are attached, your chromosomes are all lined up like metaphase, but then you forgot this one, right? That's not supposed to be there. Does that look like it's lined up and ready to be segregated? No. So what ends up happening is MAD2 will sense this unattached kinetochore, and it will bind there. And it actually inhibits CDC20 APC. So APC, remember, is activated by CDC20, so it'll inhibit that APC from working. So do you guys understand how that might prevent progression of mitosis? Who wants to explain that to everyone? Right, so there's a protein called MAD2 that binds to unattached kinetochore, so it senses chromosomes not attached to the mitotic spindle, and it also inhibits CDC20. How is it activating a checkpoint? How is it preventing progression of the cell cycle in mitosis? What's supposed to happen after this? Anaphase, right? What happens to chromosomes during anaphase? They're separated. What does APC need to do to separate chromosomes? Degrade securin to allow separase to degrade cohesin, right? So if you turn off APC, are we going to be able to proceed to anaphase? No. So that's how it works, okay? So now let's think about cancer drugs. Remember we were going through back in the cytoskeleton all these drugs that work to inhibit microtubule function in some way. A lot of them are cancer drugs. Taxol was one of them. Think back. Dig deep. So now you have to think to yourself, okay, so why do these drugs that destabilize microtubules inhibit progression to anaphase? Can you guys make that connection? Someone make the connection for me. Drugs that destabilize microtubules, they're used as cancer drugs, right? Cancer drugs are supposed to prevent cells from dividing because cancer cells divide uncontrollably. So a good cancer drug is going to slow down cell division, slow down mitosis. Some will target microtubules and they inhibit progression, metaphase, anaphase, transition. How does that work? What do you think it's doing based on what I just told you? Yeah.
Microtubules make up the spindle fibers, and if you destabilize that, then you cannot proceed, you cannot separate.
Right, so microtubules make up your mitotic spindle. So if you destabilize them, you can't proceed. Why? What's the mechanism? Yeah.
The MAD2 is going to bind to the unattached chromosomes and then inhibit the CDC20 ABC so that anaphase can't progress.
Exactly. So then MAD2 will bind to those unattached kinetochores on the chromosomes and then inhibit APC, turn off APC, separase can't be activated, cohesins can't be cleaved, chromosomes can't separate. Isn't that a nice mechanism? I love when science is so nice and neat. It's never that way. There must have been like 10 years of trouble. Actually, so I was looking this up because in my old slide I had it's believed to inhibit CDC20 APC because that's what it says in the textbook. And I thought about it. I was like, something must have changed in the last year. And so I looked it up and actually there's a nice review that was published about how this works. And because I love mechanisms, I'm just going to tell you a little bit about it. But it's really logical, actually. So MAD2, it actually recruits other proteins that then bind to this. It's a competitive inhibition. Basically, it recruits other proteins that bind to APC. So here's APC CDC20. MAD2 comes and binds, so it's kind of inhibited. And then this BUB3, don't worry about memorizing it, other complex of proteins comes and binds and it inhibits the substrate, which would be separase in this case, from being able, bless you, to bind to the ubiquitin ligase. If you can't interact with your ubiquitin ligase, you can't be ubiquitinated, you can't be degraded, and so on. And that's how it works. So there's a million papers about this. It's really interesting. Any questions? You guys all know about competitive inhibition, right? A nice, neat mechanism. Okay, so chromosomes, they separate through shortening of kinetochore microtubules and sliding of interpolar microtubules, resulting in pushing the poles apart, right? And this is just kind of what we spoke about at the end of class on Friday, right? So you've got your kinetochore microtubules that attach to your kinetochores, so those are binding to your chromosomes, and then your interpolar microtubules are the ones that bind to each other, and those slide each other, so those slide against each other, so they push the poles apart, and these shorten, and that's how you have chromosome segregation. And that's based on what? So what's happening? So what's causing the shortening of these microtubules? I heard someone whisper catastrophe, right? So you could guess that catastrophe is being increased on these microtubules by what kind of proteins? Yep, MAPs, but catastrophe factors, right? So MAPs that induce catastrophe. And then what's causing these proteins to slide against each other? Kinesins. What direction are they moving? To the minus end. To the minus end. So dyneins, right? Is that right? Or no? Maybe kinesins. No, kinesins. Kinesins. Sorry. I always get turned around with kinesins and dyneins. We all have our faults. Yeah. And so remember we talked about regulation of activity of kinesins in microtubule. Yep.
I was just wondering where like the shortening of the ones that are attached occurs. Is it like at the plus end, the minus end, or something?
So remember, it was the last slide of Friday's lecture, and it's at the plus end, and there's this collar, this protein structure that kind of is attached to the kinetochore and interacts with the microtubule, but slides down the microtubule. So as you have catastrophe occurring, this ring kind of slides down.
So it's like the stuff inside of that, past that ring that gets.
Yes, yes. So the microtubule is inside that ring, and it slides, and then you have catastrophe on the other end of it. Yep. Any questions? Is it all coming together for you guys? Are you like, wow, Dr. Ehrlich's amazing? Or are you saying, wow, she's totally stressing me out? Okay, so don't forget. So the nuclear envelope, this is just kind of a cool thing because I love microscopy. The nuclear envelope, remember, it disintegrates during mitosis, and it needs to reassemble during telophase. So if you look at the staining here, so LAP2B is green. It's a protein that interacts. It's an intermembrane protein of the nuclear envelope. So you can see during anaphase, it's like this, and then as you get to telophase, it starts to reassemble. You start to have this reassembly of your nuclear envelope. And in your nuclear envelope, you've got all these things that we talked about when we were learning about nuclear import. Remember, you've got all these nuclear pores that need to form again. And you've got RAN, and you've got all these intermembrane proteins. And yeah, it was kind of cool to see how all this reassembles again. Okay, so now we're at cytokinesis, right? That's what it looks like. Everyone like that? Okay, so during anaphase, actin and myosin 2, start to reorganize to form the contractile ring, right? So you guys know exactly how this works, right? Because we just had an exam on this. So remember, they need to relocalize. So here is your actin. Here's I should have left the light on. Turn it all off. Right, so there's your actin. So it's kind of reorganizing around that center part to form your contractile ring, and there's your myosin 2. So what mechanism is regulating this reorganization of actin? Who wants to guess? I'm not even going to tell you because you just answered a question about it on the exam. I'm just going to stand here. You guys want to talk to your friends about it for a minute? Consult each other. Consult. What is regulating contractile ring formation? Okay, I know it's really fun to forget what we just had an exam on. I know it's really hard. It's hard. All these things to remember.
Like I have a slide, I just put a slide and I hope I can't remember it.
Okay, so who wants to throw out a name of something? Okay, a lot of people are saying formins. What activates formins? Rho proteins, remember? You don't remember Rho at all? Well, you're going to remember it again in a minute. Okay. Okay, remember the Rho family of GTPases, molecular switches. That's what, remember there was the whole question about chemotaxis and movement of cells towards a chemoattractant and how you have RAC activation at one end and Rho activation at another end, and you get ARP activation and the web of actin associated with the leading edge at one end, and then you get Foreman activation and parallel contractile bundles, right? So that's what's happening here. You have a contractile ring. You hear contractile, you think Foreman's, and then you need to think of, what is it, Rho? So here's the picture. I was joking when I said I wasn't going to tell you. So activation of Rho A is what triggers contractile ring assembly. Remember, Foreman activation, right? So that's actin filament formation, and then you've got these kinases that are activated by Rho, including ROCK, which is a kinase, and that's involved in phosphorylating the light chain of myosin. Remember, that was the slide with the tail of it bent back up, and whether or not it's phosphorylated or not regulates bipolar filament formation. Right, so then you get phosphorylation and bipolar myosin 2 filament formation, motor activity. That's what does contraction, right? So then you know there's probably a GAF that's involved for activating this Rho protein because it needs to add a GTP, right? And then, of course, it's going to end up inhibiting the phosphatase because you don't want to remove your phosphate group from your myosin. You want to keep it phosphorylated so it could be active and form that bipolar filament. Questions about this? It's just review. Okay, everyone's happy. Who's not happy? Who's very, very sad today? No one? We have a sad person. Sometimes we're sad. It's okay. I'm really so I've been thinking a lot about emotions and how dismissive people are of emotions. I have children. We have to think about these things, right? So you're like when your kid starts crying about something, you're like, you're fine. That's actually a horrible thing to do to them. Because it makes them not trust their emotions, and then it's not good. So anyway, so we have to all be very honest with each other about how we're feeling. We're going to have a group hug. Everyone. Okay. Group hug at the end of the semester. For all those who passed. Who has a question? Yay! I thought you were just telling me you were sad. Okay, what's up?
I was wondering, for ring formation, what determines the volume for each, how much volume each side gets?
Oh, I love it. Volume. Like, why isn't one cell completely, huge and the other one teeny tiny? I know, I think that's an extra credit. Because I have absolutely no idea, but there's clearly a mechanism, right? Because you don't have all these uneven... Well, okay, wait a minute. Before we make it an extra credit, I know, you guys are due an extra credit, aren't you? Did you have an answer for this?
No, I was just thinking, what about when it's pushing apart? Just like when the mitotic spindle or this centrosome kind of has that turgid pressure on the plasma membrane. So what are the ones that push each other apart?
The interpolar.
Yes, when the
Spindle?
interpolar ones push each other apart, there's probably like equal force going on each side.
Okay, so something about that's true. You do have pushing of your poles of your mitotic spindle to opposite sides of the cell. But I guess the question is, and now I want to go back to this one right here. I almost want to say there's some kind of interaction, though, between the microtubules of the mitotic spindle and maybe some of the row or something, because it's all about localization, right, of your actin, right? Now, and then there's a part of me that wants to think of it like a balloon. Like if you have a balloon and you make it tight in one spot, I don't know. Let's make it an extra credit because I don't have a real answer, but I've just like given you ideas to think about. So the question is, what regulates? It's almost like what regulates localization of the contractile ring, because it's in reality that's what's going to affect the size of the resulting cells, right? Because if the contractile ring forms at the end, you're going to have a teeny tiny little vesicle and a giant cell. But then based on what Jesse said, that probably couldn't happen because of how the chromosomes are segregated to opposite ends and how the spindle poles push each other apart. Okay, so I want a mechanism of localization of contractile ring assembly, I guess. You have till noon tomorrow, and it cannot just be what you think. It needs to be referenced. People always want to tell me what they think, and it cannot be just a repeat of the question. And if you don't know, you don't know. Don't just send me something that has the words contractile ring in it, because you won't get points for that. And sometimes it's not known, and that's okay. Okay. Good question. Good question. Okay. So the next thing we need to talk about. Okay, so we're done with M phase, right? What's after M phase? G1. And then it starts all over again, right? So I put this slide up because I want us to remember that there are two types of APCs. There's the CDC20 APC that's active in M phase. But don't forget, CDC20 APC degrades M cyclin, so it turns off the MCDK. But if you still need APC though, because you need to keep MCDK levels low to go into G1. So there's this conundrum that we're going to talk about. Okay, so what did I just say? The problem is that entry into G1 requires low MCDK activity. You need to get rid of your MCDK activity or your M cyclin. Otherwise, you can't completely exit mitosis and go into G1. But since MCDK activates APC, it's going to also degrade, and MCDK and M cyclin is degraded by APC. You have this conundrum where you're actually degrading yourself, which turns off the activity of APC if you're not careful, and then you'll never get out of mitosis, right? So just a reminder to people, remember APC, CDC20 is this activator of APC, so it can go on and degrade things, and one of the things that it degrades is M cyclin to turn off MCDK. That's very important for leaving mitosis. And so in M phase, M cyclin level needs to go down. And it goes down because it activates CDC20 APC. So that turns on APC, and that degrades M cyclin, so it goes down. But then that shuts off CDC20 APC. So how do we solve this conundrum? Does everyone understand the conundrum? No. Thank you for your honesty. If you need APC to degrade M cyclin so that you can get out of mitosis, but when you degrade M cyclin, it shuts off APC because M cyclin, MCDK is what activates APC, then you have a problem because then your M cyclin is going to go back up and your MCDK is going to stay on. And you'll never leave M phase and enter G1. And you'll never be able to go through the cell cycle again. That's the problem. How are we feeling? Everyone feeling good this Monday morning? Is it Monday? Okay. So the solution is CDH1. This is the other activator of APC. It actually keeps APC active and so it trades places with CDC20 is what it does. And it's actually activated by MCDK. And so that's one thing. So you have CDH1, which takes the place of APC, keeps APC on, so it keeps M cyclin levels down, keeps MCDK off, so you can go into G1. There are a few other things. So you generally need to keep your cyclin CDK levels low to enter G1. And so these are different mechanisms for how this happens. So one is keeping APC active with another activator, CDH1. And then also you end up with increased expression of CKIs. What are CKIs? Inhibitors, right? Cyclin kinase inhibitors. So that's one thing. So you might have higher levels of some things, but you'll be able to inhibit them and keep them off. And then you also get downregulation of M cyclin transcription at the same time. So that all keeps the levels nice and low so you can exit mitosis and enter G1. And so that's what this is showing here, right? So at M phase, you've got this high M cyclin, and it needs to go down so you can enter G1. So it starts out with MCDC20, with APC CDC20, but that degrades M cyclin, and then the CDH1 APC takes over to keep those M cyclin levels low. So this is very much controlled by ubiquitin ligases. So this is from the figure I showed you, I think, last week, but I'm bringing it back because it's just important to see how these things are regulated. So at late M phase, right, CDC20 is ubiquitinated and degraded by APC. CDH1. So remember, CDH1 comes along and takes over. CDC20 goes away, so that's how you get rid of that. And then APC CDH1 degrades SCIP2. SCIP2, remember that SCF ubiquitin ligases? S stands for SCIP. So that was this SCF ubiquitin ligase. So it actually turns off SCF ubiquitin ligases. And that allows for accumulation of these CKIs. And that inhibits SCDK. Because remember, you're over here, but while you're going, even though you're only over here, you are starting to have that gradual increase of your S cyclin, but you don't want it active yet. Remember, your cyclins start to build up before they're needed. So you have this accumulation of the CKIs because you've turned off your SCF. And so then as you go from G1 into S, you get SCIP2 starts to build up again, and you degrade your CKIs, and then you activate your S cyclin CDK, and that allows and that results in turning off APC, because what it does is it induces phosphorylation of CDH1. So then APC goes down, and then that allows M cyclin to build up again, right? Because by then you're already over here, and you need to start increasing your M cyclin levels, because what's going to be after S phase? I mean G2, but really mitosis. So it's all about regulating different levels of proteins at the different times so that you can go into the correct phase of the cell cycle. Any questions about this concept? Yes?
What would you know these steps in?
Do not memorize this. Just understand it. Yep. I might say something like, I tend to give you the background information you need to critically evaluate things. So I'll never say, give me the steps of ubiquitin ligase activity throughout the whole cell cycle, because that's not useful. But it's just useful, though, to understand how these things are going up and down, and that's what's regulating the cell cycle. Questions? Comments? No, I don't want to hear any comments, actually. I don't want to know what kind of comments I would get. Okay. Okay, in the next few minutes. Okay, so a fertilized mouse egg and a fertilized human egg are the same size, right? Pretty much. We're changing. We're switching gears. Switch gears. Everyone stop. Take a deep breath. Don't forget everything. Remember it all. Let's switch gears. Okay, so you have fertilized mouse egg, fertilized human egg. They're the same size. So what factors are responsible for the huge difference in the size of an adult mouse and an adult human? Would you say an adult mouse and an adult human are the same size? Okay, good. Should that be a question on the exam?
Yes. Yes.
An essay? A 50-point essay?
Yes.
Okay. Okay. I just have to get approval from the chair for that. What do you think is responsible for that size difference? Have you ever thought about it? No, I know. Either did I before I read this textbook. But it's kind of an interesting thing to think about, right? Why doesn't the mouse grow to the size of an adult human? Ooh, bad news, right? That would be seriously bad news. That's bring new meaning to a mouse problem. Anyone? I'm not answering. This is discussion time.
It's inside of it. Are there different genes and different chromosomes inside of it?
Well, mouse. Okay, so I just heard. Are there different genes and different chromosomes? Is it what's inside that regulates things? Well, that's a very well. So the mouse and the human are quite close genetically,
Okay.
surprisingly. And yes, genes operate in different ways, but they are so close because mice are frequently used as a model for human function. Even more so, like, thank you. How come yeast and humans aren't the same size? Because yeast are really close genetically. But anyways, it's a different story. But yes, it is something that's inside. It's a regulation of something inside that does determine this. Who else was raising their hand? I saw a hand in this corner.
Does it have something to do with regulation of how many times the cells enter cell cycle?
Mm-hmm. Yep, so it has something to do with regulation of cell cycle. So of cell division, one thing. Yep. So it's kind of a balance, right? Right, because really what determines the size of things is how many cells are in it, right? If you've got two livers sitting on on a dissection tray or wherever you put livers. I guess if you're eating them, they're in the oven. Who eats liver? You do? Some people like liver? No, my parents used to eat it. Anyways, so the difference is the number of cells in your liver, really, right? That's the difference in the size. So, and the number of cells has to do with how many cell divisions you have, right? In a way. So there are a few things, right? So cell growth, so your cell size might differ. But mouse cells and human cells are relatively the same size. Cell death, right? So if you have less cell death, then you're going to have more cells, right? So survival factors that suppress apoptosis. And then cell division. If you have more cell division, you have more cells. And these are stimulated by mitogens. And we're going to just talk briefly about mitogens for the next 2 or 3 minutes. So mitogens are anything that stimulates cell growth. You can think of it that way. So they stimulate cell division by stimulating G1 CDK and G1S CDK activity. You know what? Let's stop here because this is a whole new section that we're going to go into. And then we'll start here on Wednesday. You can think more about liver.